Hi, good afternoon, everyone. So I'm Ingrid, and I'm presenting a work that's called Probing Mastoparan-like Antimicrobial Peptides and Anionic Model Membrane, Affinity through Structural and Electrostatic Analysis. So I'll divide my presentation so I can show you at first an introduction, which I'll give you some context and some justification to do this work and which system we wanted to study. Then I'll go to the structural analysis that I did based on molecular dynamics, conventional molecular dynamics simulation, and the energy landscape visualization method for the antimicrobial peptides on water and its spontaneous absorption to a lipid bilayer. Then I'll move on to the electrostatic analysis, which I did constant pH molecular dynamics with both peptides that I worked with and two different approaches, and them on aqueous solution and also adsorbed on the lipid bilayer. I'll show you some difference of charge and pKa estimation that I did for this system. So first, at the introduction, I want to talk about the system. That's the antimicrobial peptide. They overall has positive charge, are small chain, are small polypeptide chain. So it's a little small peptide chains for around 50 maximum 50 amino acid maximum, and they have weak interaction with the human the eukaryotic cell because of the lipid composition that it has, that in the outer leaflet it's predominant with deuterionic phospholipids and some cholesterol. On the other side, they have very strong interaction with bacteria membrane because of the anionic phospholipids. So we have some experimental data that we compared our computational work with. So this system is, so we work with two antimicrobial peptides that act as antibiotic agents to the bacteria bilayer. They have short chains, overall positive net charge, and they adopt a structure that's amphipathic alpha helix. I'll show you what this means shortly. And they are present on defense cells of a lot of organisms, and they act on lipid phase of the membrane, so they don't need a receptor. They act disturbing the lipid packing, so causing dead cells through the leakage of the interior of the cell, the cytoplasmic material. So the amps that we worked with are MP1, that's from Polybia paulista VASP from Brazil, and it's analogous that we call HMP1. That's the same that MP1, but we changed the lysines to histidines to have a peptide that's more responsible to pH changes and would only work on acidic pH. And these peptides, experimentally, they're non-hemolytic, non-cytotoxic, and they show this amphipathic alpha helix structure that you can see. I colored the residues, so when it is in its alpha helix structure, the polar and charged ones are in one side, and the other side is populated mostly by hydrophobic ones. So it would get inserted in the lipid bilayer, the hydrophobic ones interacting with the tails, and the polar ones with the heads of the bilayer. So we used a mixed membrane with POPC and POPG. POPC is deuterionic, POPG is negative, on a ratio to 70 POPC to 30 POPG, just like the experimental work was. And our peptide has this sequence. So you can see the only difference is the lysines were changed to histidines. So at acidic pH, it would have the same charge, but you can see that the side chain of lysine is way longer than histidine, and it could have some different on the structure that I'll show you that we see on our simulations. So I'll start with the structural analysis that we did some molecular dynamics simulation in two ways. At first, the two peptides were constructed and solvated on aqueous solution with water and salt, and some steps of energy minimization, equilibration, and the simulations were performed at different temperatures from 280 to 360. So we could estimate free energy that I'll show you later. And also we could see that higher temperatures could open the helix more fast than the lower temperatures. And we also put the peptide on a simulation box that had this mixed lipid bilayer that was previously equilibrated until the IR per lipid should be constant. And then we did 12 replicas for each peptide and observed the spontaneous adsorption. Not all the replicas I had adsorption, so I only analyzed the ones that was more close to the experimental results that I had to compare, because we had some tryptophan quenching that was not done by me, was done by a colleague. So we used her data to compare with the simulation and choose the ones that we wanted to analyze. All simulations were performed with NAMD and Charm 36 force field. And to perform the structure analysis, we use this Elvin technique, that's an energy landscape visualization method, that we want to take a simulation and a lot of confirmations that our protein had from the trajectory, and project these structures on a two-dimensional space. So it's a multi-dimensional reduction to a 2D projection. The matrix that we used was the Q-holiness matrix. That's a way to calculate dissimilarity, because you can get each structure that a pair of amino acids has on each frame of your simulation that you're projecting. And you can calculate the distance between the structure with this metric, and that sees how similar they are. And if you do 1 minus the Q-holiness, you have the dissimilarity, so how different they are. And you can project them on a 2D space. So getting this all together, this work was done with all these guys, Alexandre, who is here, Murilo and Rafael, that are colleagues from the group, and Professor Victor. And if we put our projection with the Q-holiness matrix, colored by Q-holiness matrix, the axes have no particular meaning. The important is that each dot of the projection is a structure from our simulation. And this projection is the two peptides put together on a single projection. As we used only C alpha to see the structure, because we wanted to see the backbone structure, so you can mix MP1 and HMP1 because the side chain wouldn't be a problem. And the more red ones belong to the folding basing, so it's more folded, and the blue ones are the most unfolded basing. If you separate this between the two peptides, MP1 and HMP1, you can see that MP1 populates more the folding basing than HMP1. The more red values, so the higher Q-holiness, are on a part of the projection that HMP1 does not populate, while on water. Okay, so this is our structure that comes from the simulation of the peptides on water. Here you can see it colored by density, so the density of the dots, not each dot is a structure, but a density, and how the the backbone of the peptide is folded on each of the signature populations of the projection. The purple circled ones are the ones that only MP1 populates, that has the more center part of the peptide remains folded. Our hypothesis to that is that lysine has longer side chains that could interact with the aspartic acids, doing salt bridges that could stabilize the central portion of the structure. Histidine has a more short side chain, so it will not go that far to interact by salt bridges with the aspartic acids. So the pink one is a population that has more dots on the HMP1 than MP1, but it does not belong to the full folded basing that correspond to the structure that are more active, that I'll show you later. Okay, so those are all the dots that I had on the previous projection, but separated by temperature. So you can see that MP1 at 300 Kelvin populates the folded basing. The first, I think, 20% of the simulation I took off as some kind of equilibration. So just the final part of the simulation was analyzed to not depend so much to the first initialization that I had. So the higher the temperature, more the unfolding basing got populated on both peptides. So to add to that projection, I got some frames from the simulation that I had that showed the spontaneously adsorption of both peptides to the mixed bilayer that I had, which is POPC and POPG. And if we add these frames to the projection that I had before, you can see it changed a little bit because of the method. It's an iteration method, so it's based on a first scheme methodology that randomly spread the structures on the projection and then goes moving to closer the ones that are more similar, and the ones that are different, it tends to separate. So each time you run it, you have a little bit of different projection, but you can see the same folded basing as you see before, separated by some local signatures, and the folded basing on top. If you separate by MP1 and HMP1, you can see that MP1 also populates more the folded basing. And now we have a little, it's not actually, here, very similar to the one that we had on MP1 in the folded basing, that I'll separate by only the frames that were added from the simulation that was the adsorption peptide. On pink, I have the HMP1 by density, and purple, the MP1. If you separate these clusters, you can see that HMP1 goes from a more unfolded when it's on water and it gets inserted on the lipid bilayer, it acquires like this cluster 2 structure that is only for the C-terminus is a little unfolded. The rest of the peptide is an alpha helix structure. While MP1 has a more stable alpha helix structure, it does not get that unfolded easy on the presence of the bilayer, so it remains on alpha helix most of the time. And when it gets adsorbed, I have this cluster number 3 that is the alpha helix is like perfectly placed, that is the adsorbed configuration. So the adsorption of both peptides goes a little different on the secondary structure by a little bit, but they are really close on the projection. It's important to say that experimentally, these peptides on acidic solution, MP1 is more active than HMP1. HMP1 has a a good lytic activity, but not as good as MP1. That's the one that comes from nature. The HMP1 we did on the laboratory. So those are the structure difference from these peptides. We also have some electrostatic difference that I'll show you right now. So we did the simulation in different temperatures so we could estimate free energy by WEM. And here is the projection colored by its free energy, like blue to white, from lower free energy values to higher. And the frames that were added from the adsorption simulation are colored on red when it's only close to the membrane but not interacting. The yellow ones, the peptide is interacting with the lipid bilayer, and the purple, it's fully absorbed. So you can see how it goes from water to the inside the membrane, looking for its structure. So as I told you, MP1 has a more stable structure, even with a CD experiment that we put it on the paper. It agrees with the simulation that MP1 has some structure in water, but HMP1 does not. It's go random coil. And when it's upon its adsorption to the bilayer, it gets the structure folded on a alpha helix. So just so you can show the same clusters as before. Electrostatics from these peptides we did by doing constant pH molecular dynamics simulation. I used the method that was developed by Brian Haddock and Benoit Roux, which consists on doing the simulation with a chosen protonation state for the residues, and then you choose one residue to change and do some switch that is non-equilibrium simulation. It goes by alchemical transformation to destroy or create the proton. And then if it's accepted, the simulation continues. If it's rejected, it goes from where it was before, and then the previous ionization state. This has very high computational cost. It's not able, well, 2 to 3 years later, when we're not able to see structural differences using constant pH because it's really slow. It's like 5 times lower than normal MD. So we used the as start structures what we had before. So I got the peptide on water, and I got the peptide fully absorbed on the lipid bilayer to do the constant pH from the simulation that I had done already for both peptides. So what we have here is this work that I did with Professor Alexandre too, João and Sidney, that we compared the ionization state of the residues from MP1 and HMP1. So for MP1, we let lysines and aspartic acids vary from time to time. And for HMP1, histidines and aspartic acids. So what we compared with some Guy-Chapman theory. It's some calculation that Professor João did to compare to our results. So you can see that on bulk, constant pH got on the 3 pH that we tried. We only tried these 3 because we had experimental data to compare, because I told you that's very high cost computationally. So on aqueous solution, MP1 has a charge of 2 and constant pH and HMP1 it was on acidic pH a charge 1. While in this is not good agreement with the Guy-Chapman theory, but when you look to the absorbed lipids and which charge it would have by Guy-Chapman on the surface of the bilayer, you can see that the results are most similar than the ones that we did on water. And the charge of both peptides on acidic pH is too similar experimentally. Constant pH is a little different, but also we can see that it's in the decimal, right? The 3.3 and 3.1. So we think that the difference on the lytic activity could come from the structural difference or this small electrostatic difference between these peptides. I also have some pKa estimations that I did, but as I told you, I only did on 3 pH, so I was not able to I estimate pKa for all the residues, only aspartic acids on the MP1 because lysines were always protonated on this pH. And for HMP1, we got the histidines and the aspartic acids only on adsorbate. When we think this is a small peptide, the pKa should be near the ones that this residues has on aqueous solution free. So for aspartic acid, we would expect on aqueous solution something around 4, while adsorbed on the bilayer, you can see that I got from 5.5 to even 6 of pKa, showing that the bilayer got higher pKa when it's adsorbed. So we had some conclusions that MP1 has a more stable secondary structure, and while HMP1 populates more the unfolded basin on the projection. When absorbed, both peptides populates a more folded basin, so it is the bilayer is needed for them to fold and interact, for HMP1 to fold and interact. And the constant pH showed that the residues had higher pKa while interacting with the light layer. And the acidic pH peptides are different on aqueous solution. However, they are pretty similar, roughly the same when they are adsorbed. So I'd like to thank you all, the organization and the agencies that pay our money to do the work. Thank you.
Do you have any question? So I can start. my question is quite simple. When you're estimating the pKa's that you did in the end, you took into consideration the lipids that you used. So you used POPC POPG mixture.
Yes, but we did not change the lipid ionization state, just the proteins.
But okay. When you did the calculation,
yes,
the lipids were there.
Yes, they were
explicitly.
There. Yes.
Okay, thanks.
I have one question regarding this analysis method. How is it called? Elvin.
Mm-hmm.
When you analyze the basins, have you checked whether the basins are homogeneous or not in terms of conformations? Because with other methods, my experience is because of the reduction of dimensionality, mm-hmm. you quite often end up with basins that can be highly heterogeneous. Mm-hmm. If you just look around the minimum, maybe they look homogeneous, but sometimes they are not at all. Mm-hmm. Have you checked that?
Yeah, I did this. We looked. Mm-hmm. Let me see. We look the density of the region, so you can see if there were more or less structures, and we took some structure that are near, and if you can see it in this figure, there are a lot of superimposed. So you can see they are pretty similar.
Yeah, that's good.
Any more questions? No. Thank you very much.